So, today we will actually talk about the scaffolds. So, we looked at the three different arms of the tissue engineering triad, right. So, we saw materials, cells and signals. So, we looked at a brief introduction of all of these things. So, today we will start with the aspect which I work on, which is the designing of scaffolds. So, we will talk about different materials that can be used for scaffolds and what are the desired properties and so on. So, the first aspect which we are going to talk about is the first type of scaffold we are going to talk about is the extracellular matrix itself. So, as you know, ECM is present all through your body, and that is where the cells actually attach and grow. So, there are two aspects to understanding this to be gained from this lecture. First is to understand what is the ECM and actually how it is relevant for tissue engineering, right. So, for us first we need to know what are the components of ECM, how it is structured and so on, so that we can try to mimic it. So, that is a crucial aspect which we need to start with, and then we will talk about how ECM is currently being employed in tissue engineering, and there will also be a small reading assignment, so which is just a research article which you would have to read, which talks about using extracellular matrix for tissue engineering applications. Okay. So, before we go into details of extracellular matrix itself, what are scaffolds and what are their roles? Scaffolds have a very critical role in tissue engineering because they actually can direct the growth of cells. It could be either the cells which you have already seeded. So, you could either place the cell seeded on the scaffolds and use this cell seeded scaffold as a tissue engineered construct, or you could use only the scaffold and you might have cells migrating towards this or migrating away from this, right. So, it can regulate these factors. So, mammalian cells, as I had already mentioned, are anchorage dependent, they are actually adherent cells. So, they need some substrate on which they can attach and then grow. So, scaffolds are the matrices which provides this type of a surface, and they also provide the mechanical support for the tissue. So, depending on the tissue, the mechanical properties and other physical properties can be imparted by the scaffold. So, the cells which are delivered, you might want high loading, and you might want specific cell attachment to the sites of which you are interested in. So, for these types of things you need a scaffold which needs to be engineered properly. So, having said this, what are the desired properties of a scaffold? Can you think of properties which you would want in a scaffold? I will write down as you shout out the answers you think of. Okay.
Mechanical properties.
Okay, compatibility, mechanical properties.
Biodegradable ability.
Okay. Biodegradability. Okay. So, you had something.
Same.
Okay. Okay. What else?
It shouldn't be immunogenic.
Okay. Part of compatibility. Okay.
High cell adherence.
Okay. Cell adhesion. Okay.
High surface area.
Porosity. Okay. Why porosity?
Nutrients.
So, the other ones are more direct, we know why, but porosity, why?
Because we'll be adding molecules that should diffuse and reach the tissue.
Okay. So, it will help in diffusion, that is one reason. Can you think of any other reason?
Nutrients.
Cells will infiltrate.
Oxygen.
Oxygen and nutrients.
Oxygen transfer. Okay.
Toxic byproducts of cells
Okay.
should also go.
So, all these are diffusion. So, diffusion in and out, that is one aspect, and then cells in infiltration is one aspect. Okay. Porosity also will give you the real structure of an ECM. Okay. It is closer to what an ECM would be. ECM has these porous matrices in which the cells can actually adhere and grow. Okay. Anything else?
Non-toxic.
Again, part of compatibility. Okay. Is that all? Can you think of anything else?
I don't know word for it, but like there is a possibility that the scaffolds can be infected by bacteria or foreign pathogens. So
Okay.
that property, I don't know what the word.
Okay. So, you want the scaffolds to be
Not have-
okay, to be bacteriostatic or bactericidal.
Yes.
Okay. So, that could be a desired property, but again that can be part of compatibility, right. So, again it is Just like how your own cells rejecting it, again having some infections can also come as part of compatibility, but you can list it as a separate property as well. So we'll just call it antibacterial. How that antibacterial property is given can be discussed, and it might also depend on what type of tissue you are talking about, right? So if you are talking about wound dressing, which is for skins, then you might want a very antibacterial material, whereas if it's going to be for something else, it might not be that crucial. Okay.
Interaction with growth factors, like adherence of growth factors and things, ECM is also involved in that.
Okay, so ability to deliver signals, can I say it that way? Okay, signal delivery. Anything else?
Lightweight.
Lightweight. Okay, why? So it's
So that if it is implanted, then it won't restrict our movement or something.
okay. So I would say it needs to be of similar density as your natural tissue. So saying lightweight is probably not the best way to describe it, because see, if you are going to talk about a bone tissue, that's obviously going to be heavier than soft tissue, and weight is not the parameter. You are looking at density, which is a more uniform. So what is density compared to? So what's the fundamental difference from a property standpoint between density and weight?
Weight is volume based.
Yeah.
What if there is a property? Intensive properties and extensive properties. Okay, intensive properties are ones which depend on size, which don't depend on size, and extensive properties depend on size. So when you're talking about something like this, you're talking about the property of the material itself, not it should not vary with the size, right? So obviously if you're replacing a large tissue, it's going to be heavier anyways. Okay, what else? Okay, so let's look at what I have. So many important things you have given. So support and deliver cells, so induce, differentiate and channel tissue growth, target cell adhesion substrate and stimulate cellular responses would all come under providing signals. So these are just the different signals which we are looking at. Biocompatible, biodegradable. So again, compatibility is crucial because you need to make sure there are no immunogenic responses and so on. Degradable depends on the type of application. So how fast it needs to degrade or slow it needs to degrade will depend on the rate of regeneration of the tissue we are talking about. And then you have to talk about the large surface to volume ratio, which is one of the aspects which porosity will provide you. And when you are actually having this large surface to volume ratio, for the same volume of implant, you can actually have a lot of surface on which the cells can attach, right? The cells are only seeing surfaces, they are not going to see the scaffold as a whole, right? So when you provide a lot of pores, or if you make it into nanofibrous matrices, then you have a lot of surface area for the same volume. So this will help the cell adhesion and growth. Mechanically strong, structurally stable. So these are all the properties which we think of from a biological standpoint. From a final production and manufacturing standpoint, we need the material to be processable and malleable, and it should also be sterilizable. So there are different techniques which you can use to sterilize, but whatever technique you use should not affect the properties of the material, right? So if you are going to use a protein on top of these scaffolds, or like a collagen, it should not get denatured while you are processing, while you are sterilizing it. So these are important factors which you need to account for, and choosing the technique used for sterilization would also matter, and you can actually design things appropriately. Okay. So these are the desired properties you are looking for. How you provide these desired properties is the challenge. So you need to look at what materials can be used, what chemistries can be used for cross-linking. So depending on the level of cross-linking, your mechanical strength can actually vary and so on.
Might be a silly question, but do optical properties matter when it comes to scaffolds?
Depends on which scaffold, right? So optical property of a scaffold would matter if you are trying to engineer a cornea, right? If you are going to engineer a liver, I don't really think optical properties matter. If you are going to engineer a nerve tissue, then your electrical properties would matter. So depending on the tissue, you would have to design the properties. The physical properties will primarily be driven by the type of tissue you are trying to engineer. Okay, mechanical properties, bones are going to have different properties, muscles are going to have different properties, and so on. So, what is the role of scaffolds in vivo? So, it helps in constructive remodeling of functional tissue, engineered tissue. So, the scaffold has to degrade and cells have to adhere, migrate, proliferate and differentiate. 3D organization of the site to form appropriate tissue should happen, and it should also help in vascularization. So, this is what would happen when a scaffold is placed in vivo ideally, right. So, if these things happen, you know that the scaffold is doing its job. So, what factors will control these processes? Blood supply, pH, oxygen, carbon dioxide concentration, mechanical stresses, host surface interaction are all some of the parameters which can actually regulate these things. So, for getting these type of scaffolds, people use different materials. So, we will talk about some of the major classes of materials, and we will also look at how materials can be processed to get these types of materials. So, first today we will talk about extracellular matrix. So, extracellular matrix would be the gold standard, because that is what is there in your body, right. So, if you can get the extracellular matrix to do its job, if you can use that directly for tissue engineering, that is what you would do. But what would be the limitation of doing that? So, if I want to use the extracellular matrix, what do you think would be the limitation? Availability is one problem, okay. Can you think of something else? Compatibility, why?
For immunogenic agents, you have to ensure that it's compatible with the person you're putting. In other words, you'll get elicit an immune response.
Okay, so that is true for cellular ECMs. When you decellularize it, it would not be a problem. That is what I wanted to get to. So, if you actually have removed all the cells and cell debris, then the matrix itself will actually not be immunogenic, because all these proteins are reasonably well conserved. So, they are conserved across species, okay. So, it will not cause any problems.
How will this be developed in vivo?
What do you
In
mean?
vitro.
In
How
vitro or
will- yeah,
vivo?
In vitro.
In vitro. How would you create a decellularized ECM? So, there are techniques for it. So, the last few slides on this lecture will be that, we will talk about it, and the reading material will actually talk about that extensively. Okay. So, ECM function is to support cells, regulate polarity, cell division, adhesion, motility. It is also involved in tissue development through cell migration, differentiation and growth factor delivery. Okay. So, ECM features which we need to understand are, they are stable with the ability to be reorganized, right. So, extracellular matrix can actually get reorganized by activation, by action of enzymes like metalloproteases. We will talk about them, and we also need to understand that the features of the ECM are going to be different for the different tissues. It is not going to be the same, even though we just call it as collagen, there are actually different types of collagen, and which collagen is present in which part of the tissue matters. So, ECM composition can be broken down to 3 major classes. So, basically 2 major classes, proteins and proteoglycans. So, the amongst proteins there are actually 2 things. One is a structural protein which provides the support for the tissues to grow, and the other are specialized proteins which have special biological functions. So, the structural proteins also have some biological functions, but we will for simplicity we will classify them as only structural proteins. And proteoglycans are basically molecules where you have a protein core to which long glycosaminoglycans are attached. So, these are complex high molecular weight components present in the ECM. They provide the viscosity and the like fluid like nature of the ECM itself. Okay. So, amongst the structural proteins, the common ones are collagen, elastin and fibrillins, and among specialized proteins, the common ones which you see are fibronectin and laminin. Okay. So, we will talk about these in a little more detail. So, collagen is the most abundant protein by weight. Okay. So, when you are asked this question, please remember this, because this is a very common question which is asked in many places, where you say what is the most common protein in the body, and inevitably most people say hemoglobin, which is not correct. Okay. The most common protein in your body is, most abundant protein in your body is collagen. Okay. This is the structural component, it provides the support, it also can have some functional properties. So, there are actually 46 different collagen genes in the human genome, and it generates 28 different types of collagen fibrils. So, these are just identified using Roman numerals. So, you would have type 1, type 2, type 3 and so on. So, the collagen fibrils are the ones which provide the strength. So, the fibrils assemble to form collagen fibers, and these bundles which are the collagen fibers actually impart the mechanical strength. So, they are very strong structures. Types 1, 2 and 3 are the most abundant, amongst which type 1 is by far the most abundant. Type 1 actually constitutes of about 90% of all the collagen in the human body. So, primarily type 1 is present in all over in all the tissues. So, these are predominantly synthesized by fibroblasts. So, if you culture fibroblasts and you maintain certain environment, you can actually make these fibroblasts secrete collagen. So, there are labs which actually do that, and our own institute, Professor Verma's lab does that. So, they culture fibroblasts to secrete collagen, and the yield of it is always a problem. So, it depends on the culture condition. You would not know whether how much of a matrix you would get. Okay. So, this is one way to get it, get the ECM type of a scaffold. So, epithelial cells can also synthesize some of ECM collagen. So, as I said, there are 28 different types of collagen fibrils, and these are some of the major ones. So, type 1 collagen is seen in skin, tendon, vascular, ligature, organs, bones. So, this is the main component in bones. Okay. So, collagen component of bone is primarily type 1, and you have type 2 collagen which is mainly seen in cartilage, and type 3 collagen is seen in reticular fibers along with type 1, and type 4 is seen in the basement membrane. So, basement membrane is the one which separates the tissue from the matrix. So, you would have that in the basement membrane, and type 5 is seen in hair and nail. Okay. So, they have different mechanical properties, they actually have significantly different physical properties, and they are seen in different tissues. So, this is what a collagen fibril looks like. So, this is a triple helix which you see. So, this is a mature type 1 collagen, and so basically what happens is they are initially synthesized as pre-pro proteins. So, a pro protein is one which basically a protein precursor. When these come along with the signal peptide, they are called pre-pro proteins. So, you have pre-pro collagen which is usually synthesized. So, from this there are actually a lot of co and post translational modifications which happen for the collagen to form the fibrils. So, collagen monomers, which are called the alpha chains, self associate into triple helical structure. So, collagen has a triple helical structure basically which are called the collagen fibrils, contain two identical alpha chains and a third alpha chain which is different. Okay. So, the type 1 collagen itself is encoded by collagen Col1A1 and Col1A2. So, there are actually two alpha helices from Col1A1 and one from Col1A2. Okay. So, these three form the triple helix which is shown here. So, the two blue ones are the collagen 1 alpha 1 and the red one is the collagen 1 alpha 2. So, from this they form the triple helix which forms the collagen type 1 fibril. So, this process actually shows the synthesis of collagen. So, what you have is collagen fibers are produced from a pro alpha chain. So, this pro alpha chain then, so some of it is done intracellularly and some of it is done extracellularly. So, the ones you see after secretion, after this point type, after the 6th step is the all extracellular. So, until then it is intracellular. So, you have synthesis of pro alpha chains which is the first step, and then you have hydroxylation of selected prolines and lysines. So, collagen primarily has glycine as the amino acid, and the next to that is proline and hydroxyproline. Okay. So, you have hydroxylation of certain prolines and lysines. So, the prolines are actually hydroxylated and so are the lysines. Some lysines are hydroxylated, and this hydroxylysines are then glycosylated. So, once these glycosylated hydroxylysines are present, you have self assembly of the three pro alpha chains to form a pro-collagen triple helix. So, this pro-collagen triple helix is secreted and outside what happens is the propeptides are cleaved and finally you have a self assembly to form the fibril. So, these fibrils are about 10 to 300 nanometers thick. So, what happens is these individual collagen molecules, the triple helix molecules are cross linked using by the activation of an enzyme. So, okay. Anyway, so by the action of an enzyme you have a cross linking which is done. Depending on the cross linking you would actually have the strength. Okay. So, once more and more collagen triple helices are cross linked, you would have very thick fibrils and these fibrils all aggregate to form a collagen fiber. So, this collagen fiber is what forms the matrix. Okay. So, this is a typical process for type 1 collagen and you can actually find similar processes for other collagens as well. Okay. So, depending on the initial pre-protein, so initial chain alpha chains which are used, you would get different types of collagen. Okay. So, this is from basic cell biology. So, you do not need to know the process or the biosynthetic pathways of collagen. However, what you might want to know is what are the molecules which are the precursors for collagen. Okay. So, if you have that you could probably try to use them for your tissue engineering applications or you can try to understand some of the pathway where you can push towards the generation of collagen. So, you could try in vivo secretion of collagen as a way of regenerating tissues. Okay. So, elastins and fibrillins are found in tissues that undergo significant stretching or bending as the name suggests they have elastic properties. So, examples would be large arteries, lungs and skin. So, these actually go through a lot of the stretching and bending, right. So, some physical mechanical stresses are experienced by these. So, you would have a lot of elastin or fibrillins. So, this is they are found as specialized types of fibrils which are called as elastic fibers. The elastic fibers contain large masses of cross linked elastin interspersed with fibrillins. So, the elastic fibers primarily contain elastin and there is some amount of fibrillins as well. So, elastin is basically synthesized as a precursor called tropoelastin and tropoelastin has two major types of alternating domains. One is a hydrophilic domain which is rich in lysine and alanine and the other is a hydrophobic domain which is rich in valine, proline and glycine. So, the hydrophobic domain provides the elasticity. So, if you are going to design polypeptides to prepare scaffolds, then providing these types of amino acids, using these types of amino acids will give you the elastic property which you are looking for. So, tropoelastin is expressed and then secreted as mature protein into the ECM and after secretion and alignment with the ECM, elastin monomers are cross linked and so this 3 lysine derived aldehydes cross link with an unmodified lysine to form a tetrafunctional structure which is called as the desmosine. So, this is the elastin fiber which you would have and the other major component in the elastic fiber is the fibrillin. So, there are actually 3 fibrillin genes in humans and fibrillin 1 is the most abundant and it serves as the scaffold for elastic fibers after cross linking with the elastin itself. So, this gene expression is consistent with the role in the ECM. So, FBN1 which is fibrillin 1 is secreted mostly by cells from mesenchymal origin. It is seen a lot in bones. FBN2 is secreted highest in fetal cells whereas FBN3 is expressed in embryonic and fetal tissues. Some of these are not seen very commonly in adult tissues. So, those were the structural components. So, you also had functional proteins, so couple of functional proteins which we had mentioned were fibronectin and laminin. So, fibronectin is a major component of fibrillar glycoprotein in the ECM. It has a role in attaching cells to all matrices except for collagen type 4. Type 4 when you have a type 4 matrix, laminin is involved in the cell adhesion. So, what happens is these have a multimodular structure with 3 different amino acid repeat domains, and these are called FN1, FN2 and FN3, and the primary amino acid sequence that binds to the integrin which is expressed on the cells is the RGD domain. So, arginine, glycine and aspartate. So, the fibronectin actually provides this type of a motif on which the cells can actually attach. So, that is why fibronectin is even used for coating of cell culture plates and other things, so where you can ensure cells are attached to the surface. Okay. So, people also use RGD domains. So, you can create these tripeptides and use them along with your scaffolds to promote cell adhesion. If you do not, if you cannot use fibronectin to get like all of fibronectin, you can create these short peptides which can be used, and that will help in cell adhesion. So, fibronectin exists as soluble protein and insoluble protein. Soluble protein is present in the blood plasma. It is involved in blood clotting process and links to fibrin during that process. You have insoluble protein in the ECM, where ECM fibronectin actually has the polypeptide segments which alters the morphology and helps in the cell attachment. So, you have laminins which are another set of glycoproteins. They constitute the structural scaffolding for all basement membranes. So, if you remember, we also said that collagen type 4 is involved in basement membranes, right. So, along with collagen type 4 you would have laminin existing, so that it can help in cell attachment. This is a very critical component of the ECM because it has a lot of functions. It can actually bind with the integrins and other receptors, many other receptors, and it is involved in cell differentiation, cell movement, shape of the cell and promotion of cell survival even. So, because of this it plays a crucial role, and it is actually present in reasonable abundance. It is a heterotrimeric protein that contains an alpha chain, a beta chain and a gamma chain. So, which is what is shown here. So, it can also be called as beta 1 chains and beta 2 chains, which is the traditional way it was represented. So, there are at least 15 different types of heterotrimers that have been identified. The common structural features which you would see are having a tandem distribution of globular and rod like and coiled domains. So, that is what is a laminin, and this is the general representation of the laminin structure. So, other than these we also have proteoglycans and glycosaminoglycans. So, glycosaminoglycans are the most abundant heteropolysaccharides which are seen, and they are basically long unbranched polysaccharides with repeating disaccharide units. So, instead of having a random chain you would have disaccharides which are repeated, and then these are highly negatively charged with extended conformation that can impart viscosity. They also show very low compressibility. So, because of these properties they are used as lubricating agents in the joints. So, you would see these materials like hyaluronan and so on, chondroitin sulfate in the joints, and these all of these occur in different tissues. You can actually look it up on the net and you would see that there are tables which explain where these can be found. So, some of the common glycosaminoglycans are hyaluronic acid, dermatan sulfate, chondroitin sulfate, heparin sulfate and keratan sulfate. So, these are all commonly seen in your body. So, proteoglycans are basically glycosaminoglycans that are linked to core proteins which are rich in serine and threonine. They make sure that the fluidity of the tissue is maintained, and they provide resistance to compressive forces. So, they play a crucial role in making sure the tissue maintains the gel like structure gel like feature which it has. So, when we talk about ECM, we also need to understand that ECM is not a static matrix, it is getting reorganized. So, you can have proteolytic degradation to remove the ECM. So, some of the enzymes which are involved in this are matrix metalloproteases and serine proteases. So, they will degrade the tissue, and as I already said, many different types of cells are involved in secreting these ECM matrices to actually continuously remodel these tissues. Okay. So, when we talk about ECM in tissue engineering, what people are doing is people are harvesting ECM for tissue engineering applications. So, they take the tissue and so either it can be allogenic or even xenogenic ECMs, and they decellularize it, and it is well tolerated by human hosts because ECM components are well conserved. So, decellularization is very crucial because the cellular antigens are actually foreign. Even if you get it from an allogenic source, you would have to make sure that there is proper tissue type matching if you are going to have the cells along with it. So, if there is no cells, then it cannot trigger immune responses or inflammatory responses. So, for that reason you have to carefully decellularize it. There are many techniques to do it to remove all the cells. So, decellularized ECM can actually be an off the shelf product. So, this can provide a favorable environment for constructive remodeling. These can be seeded with autologous cells before implanting if you want. So, then you have a patient specific personalized medicine, right. So, because ECM is common for everybody, then you do not have to worry about that. Now, the cells which can create the immunogenic response are from the patient himself, which means there is no risk of rejection. So, you can tailor it that way. So, once you have it inside, then in vivo environment will determine the remodeling of the ECM. So, people have done in vitro experiments to study the effect of the possible stresses such decellularized ECM can go through. So, there are different techniques to do it, but what is the fundamental goal of decellularization? What do you want to accomplish? I said why we want to accomplish that, but what do you want to accomplish? What are all the things which you want to remove when you are talking about decellularization? Okay, so you want to prevent any immune response. For doing that, what are all that you should remove? Like removing cells is the general thing, but there can
Cell receptors.
cell receptors. Okay, so antibodies. So any cell debris. Okay. So, that is actually what you want to do, but specifically what you try to do is you want to remove all cellular and nuclear material. While you are doing this, you want to maintain the composition and mechanical properties and biological activity of the ECM. So, that is the challenge. So, removing all cells is not too difficult. I can always take an ECM and dip it in sulfuric acid, right. So, the idea is to remove the cells without damaging the ECM. So, how you go about doing that is the processing part which makes it unique. So, there are a combination of mechanical, physical and enzymatic processes which are done. So, mechanical process would be the simple delamination of layers. So, this is nothing but just stripping of layers. So, you take out one layer and then strip out the first layer which might contain too many cells, and you take that out, and then you can have physical things like sonication, freezing and thawing and so on, right. So, sonication will destroy the cells, will just rupture the cells, and you can freezing and thawing can also do the same, have the same effect. So, these are just cell disruption techniques, and then you can use enzymatic treatment like trypsin. So, trypsin can remove the cells from their adhesion sites, and trypsinization is done even when you do cell culture, right. So, you can do trypsin treatment. And then you can also use chemical treatments like detergents and ionic solutions. So, you need to be careful with what exactly you are doing and how exactly you are processing it. So, mechanical process of delamination needs to be done with reasonable care. So, it could just be a physical brushing of things, could be as simple as that, people actually do that, but it could also be a lot more complicated with designing well controlled equipment. So, it depends on how you want to design the process. So, these are just general techniques. So, we will go you can go into the great details of the techniques when you read the reading assignment, because if I were to describe materials and methods for each of these, it might be too much information. Okay. So, these are some of the commercial ECM based products which are currently available. Graft Jacket is a product where human dermal collagen matrix is used. So, this provides supplemental support and protection and reinforcement for tendon and ligamentous tissue. So, this is also used for covering the periosteal patch, and it acts as a protection and support for bone and tendons during foot, ankle and hand surgery. So, this is just collagen matrix which is cross-linked. So, they take the human dermal from skin, they take the collagen and they create a cross-linked matrix which can be used for these applications. Tissue mend is from fetal bovine skin, and this is also again used for musculotendinous defects. Zimmer is a collagen repair matrix which is an acellular scaffold of collagen and elastin derived from porcine dermal tissue. So, there is a
Does ECM from other animals cause immune reaction?
So, as I was saying, these proteins are very well conserved. Okay, so they do not cause any immune reactions mostly. As long as you remove all the cells and the cell debris, the chance of immune responses are very low. So, that is why people work, most of the times when people work with collagen, it is not that they are using human collagen, right. So, if you go back and look at literature, people use collagen from so many different sources. It could be from fish, it could be from rats, it could be from chicken, and just take it from anywhere and you can start using it from bovine and so on, right. So, because it's collagen is collagen, it's just a protein and it's reasonably well conserved to ensure there is no effect. And as far as the glycosaminoglycans, these are carbohydrates, right. These are just molecules which are going to have the same structure, does not matter where it comes from. So, because of this reason, you can actually use it from xenogenic sources as well. So, Zimmer collagen patch is from pigs and so is Permacol. So, this is again a decellularized cross-linked porcine implant which is being used for different applications. So, these are all some of the commercial products, and as far as Graft Jacket goes, they actually have a nice representation of what it actually how they prepare it and what actually they expect to have. So, they take a human tissue which is basically the dermal tissue here, and they treat it with they remove all the cellular components and retain collagen, elastin and the proteoglycans and make sure that the matrix is intact. So, this creates the Graft Jacket products which they commercialize, and this is by some Wright, this is the company's name. So, this reduces the rejection response because the cellular components are gone. This will also help in revascularization and cellular repopulation because you have all the pores and all the structures necessary for vascular network to be created. So, this will also have a minimized inflammatory response based on primate studies that they have shown. So, this is and they hope this will regenerate normal tissue. So, this is the commercial product which is available for different applications. So, with that we come to the conclusion for this lecture on this topic. So, this is the reading assignment which I was talking about. So, there are actually multiple of this kind of work which has been done. So, here what they have done is regeneration and experimental orthotopic transplantation of a bioengineered kidney. So, they have taken a kidney and decellularized it and seeded it again to create a functional kidney, which they have placed it in a different position. That is what orthotopic transplantation is. So, so they keep it in a different position and then see how exactly the kidney functions. So, they have done it in vivo small animal studies in this paper, I believe. So, go back and read this paper. There are many other papers as well. So, this is a paper where people have done for the entire organ, unlike just a small patch or something. So, the other commercial materials are just patches. You take the skin and you take the collagen, cross-link it and use it as a patch, right. So, unlike that, this is slightly different, okay.